I send you all my greetings. I am very sorry not to be with you here today. This is because of a health issue which I underestimated in terms of its ability to impede my travel. I want to thank IPOPI for the opportunity to speak about this subject today. So I'm going to talk about the best possible immunoglobulin product according to our knowledge. And of course, there isn't really a best possible immunoglobulin product, but we have many products as a result of the development of this form of therapy over all the years since it was initiated. I can assure you at the very outset that the products available and approved in the markets of the developed countries today, which have been overseen by the regulatory authorities in these countries, are all absolutely safe and efficacious in the ways which we shall see during this talk. This slide simply summarizes the development of these products over the decades, starting from immunoglobulins, which were only suitable for intramuscular use, going through modifications of the manufacturing process, introducing viral activation, and culminating in the products which we have today. All of these I shall discuss in some more detail. But first of all, I always like to give some idea of the history and to recognize those who have contributed to our availability of these products. And we, of course, initiate our recognition with Professor Edwin Cohn, who developed plasma fractionation with his team in the 1930s and 1940s. Cohn's original scheme was not intended to manufacture immunoglobulin from plasma, but was intended to lead to a path for albumin solution. His colleague, Dr. Ronkley, took up the scheme later on after the development of immunoglobulin, and in the late 1940s developed the scheme which you can see today, utilizing still ethanol to precipitate out impurities and other proteins, and to lead to the final immunoglobulin fraction to 1+2, which includes immunoglobulin. I do this to recognize these important people. I want to state at the outset that nowadays very few companies still fractionate immunoglobulin from plasma using this method. We have had the development of more modern methods for fractionation, still initiating with ethanol, but continuing then with other purification processes, some of which I shall show on the slide later on in this talk. This is the structure of the immunoglobulin molecule. I want to emphasize this because it has a huge bearing on what makes a good immunoglobulin. For all intents and purposes for this talk, we assess the structure of immunoglobulin through two parts. The FAB part, in which we have parts of the immunoglobulin which actually recognize foreign bodies, which we are used to characterizing as antigens, in a way that the immune system can then process them to not result in pathologies and sickness in the body. The other part, the FC domain, is what is actually involved in this further processing, and it is extremely important because it does result in the immune system, after recognizing the foreign body, being able to assimilate it and destroy it. We shall see some of these processes. It's important for us to recognize that human immunoglobulin products as used to treat immunodeficient patients are a mixture of so-called polyclonal antibodies. This means that they are derived from many thousands of individual plasma donations, which of course come from individual plasma donors, so that the whole spectrum, we might call it, of immune possibilities which are challenging the body in terms of antigens from foreign organisms such as viruses and bacteria are represented in the population. This is in sharp contrast to monoclonal mixtures of antibodies. In fact, the word mixtures in terms of monoclonal is not appropriate because monoclonal antibodies represent only one variation to the theme. They represent only one antigen recognizing site. And this, of course, is extremely important in relation to the kind of preparations which we need to treat patients with immune deficiency. We need polyclonal antibodies and not monoclonal antibodies. Of course, it is quite possible to treat patients with monoclonal antibodies if we have many hundreds of thousands of these antibodies mixed together, because each one would then be able to represent one specificity, one antigen binding site. So far, this possibility, although it is in the minds of many researchers, is not yet realized for products to treat patients with immune deficiency. I want to talk a little bit about this slide because it represents again important functionality in terms of an immunoglobulin preparation, because this focuses particularly on the FC part, which, as I told you in the previous slide, is what is involved after the recognition of the foreign antigen by the antigen binding site. This FC part, representing the constant region, is then what is used to destroy through the complement pathway the foreign organism. So when we talk about complement pathway and complement activation, we are actually talking about something which is entirely physiological and extremely important in making sure that immunoglobulin can carry out its function. Where this does not go according to plan is when it is activated in the absence, so to speak, of the need to do so through pathological mechanisms or through having antibody preparations which are not, strictly speaking, entirely molecularly intact, but which have parts of the immunoglobulin constant region attached to each other in such a way as to have what we call dimers and polymers, or aggregates. This is something which we seek to avoid because these aggregates can cause complement to be activated where it is not needed, what we call systemic complement activation, and this results in very severe reactions in the body. The first immunoglobulin preparations were such as to contain quite a large percentage of these aggregates, and therefore they could not be infused intravenously because they would result in this systemic, unwanted complement activation, leading to the severe reactions which were observed in these patients when these preparations were first tried out. And you can see here in early immunoglobulin products that the percentage which could cause complement inactivation or activation in the other sense was quite high. Nowadays, immunoglobulin products, when we measure them for this parameter, have a specification related to this which cannot be surpassed, and I shall mention this later on. But this is just to show you that the very first products did have this problem, did have this impurity, and this resulted in systemic complement activation. And we can see that nowadays we have the ability to detect these aggregates, and these are simply some demonstrations using chromatography of this, and we can see, for example, in the unbroken line in these two curves, you can see an immunoglobulin product which has a certain amount of monomeric immunoglobulin, some dimeric immunoglobulin, but hardly any polymeric or aggregated immunoglobulin. And this is in sharp contrast to the product demonstrated by the dashed curve, where you can see towards the right-hand side of the graph a considerable percentage of polymeric immunoglobulin, indicating that this product is not suitable for patients and intravenous use. This is an important observation showing the effect of pH on this phenomenon of aggregation. And the point we seek to make here is that at what we call physiological normal pH, the immunoglobulin solution does contain a significant amount of aggregated immunoglobulin, but when we change the pH to an acidic pH 4.5, we find that this is significantly suppressed and, in fact, practically eliminated. And therefore, most immunoglobulin products today are formulated during manufacture to result in a final solution which has a pH of 4.5, to result in an absence of polymeric unnecessary immunoglobulin. All these parameters which are used to specify and to ensure that immunoglobulin is of a high quality are described in the regulatory literature published by the oversight authorities such as the US FDA and the European Medicines Agency, which draw upon specification standards such as the European Pharmacopoeia, shown here on this slide, to draw upon the necessary parameters and the tests used to allow us to specify and describe an optimal and acceptable preparation of immunoglobulin. I shall go over parts of this pharmacopoeia monograph for intravenous immunoglobulin to illustrate some of these points. If we first of all look at the Pharmacopoeia monograph, it says that normal immunoglobulin for intravenous administration has to be prepared from pooled plasma of not less than 1,000 donors. And this relates entirely to the point which I made previously, that this plasma has to represent a polyclonal preparation of immunoglobulin so as to ensure that we have the spectrum of antibody specificities which we need to be able to treat patients. So this is specified in the regulatory literature and companies must conform to it. Continuing, very importantly, I mentioned briefly in the second slide that one of the developments in the preparation over historical years of manufacture of immunoglobulin has been the introduction of inactivation of viruses and other pathogenic organisms. We know that this has been a problem with immunoglobulins and with patients with immune deficiency, even though the situation was much more dramatically shown with patients with hemophilia. This slide summarizes the various incidents and various occurrences of viral transmission by immunoglobulin preparations, particularly hepatitis C, which occurred in the 1980s and the 1990s. And therefore now we have specified again within the European Pharmacopoeia and other regulatory documentation the requirement that the method of preparation includes steps which have been shown to remove or inactivate viruses and other organisms. This is a slide which is somewhat excessively busy, but I seek to make 2 points from this slide. This shows some currently available on the market, that is, intravenous immunoglobulin preparations from various companies. There are several others, but I could only fit to some level of legibility these schemes from 4 products. Most of these products, I think, are well known to you. 2 points. The first point is what I made earlier on, that although initially immunoglobulin was prepared entirely by ethanol precipitation as developed by Cohn and Oakley, nowadays, as you can see integrated within these manufacturing schemes, we have other purification processes. I emphasize this point because there are people who claim that the plasma industry has not changed its methods over the past 80 years since its inception, and that it is antiquated and non-innovative. This shows that this is entirely not the case. The industry seeks to improve its methods on a continuous basis, and we have nowadays purification for immunoglobulins, which includes methods such as chromatography and filtration, which allow us very high levels of purity in these products. The second point relates to the issue of viral activation, and where you see block arrows pointing out to steps in the manufacture, these are specific viral inactivation steps integrated, systematically and deliberately introduced by manufacturers, this is an important point, into the manufacturing process in order to remove and inactivate pathogenic agents. These steps are by no means comprehensive. There are several others which I have not illustrated in order not to have too much crowdedness in the slide. In terms of the point which we have made before, that we must have an immunoglobulin preparation which is as much as possible only representing the monomeric single molecular form and avoiding the inclusion and contamination by aggregated immunoglobulin. This is also specified in the pharmacopoeia, and it specifies size exclusion chromatography, which is a form of chromatography which separates molecules on the basis of how large they are on their slide, on their size. And you can see on the graph, which we have shown before, that we have two immunoglobulin preparations. The solid line shows a lack of polymeric aggregates. The dashed curve shows presence, significant presence of polymeric aggregates, and therefore curve 1 with the solid line passes as an immunoglobulin product. Curve 2 is a failure. This is an important test which is done on each and every batch released for treating patients. And of course, associated, as I said before, with the presence or absence of aggregates, we have the question of whether these aggregates or the preparation itself can activate complement outside of its natural function. And we have specified again in the pharmacopoeial monograph the need to detect and measure anticomplementary activity. And this slide simply shows some preparations in which we are assessing this level of anticomplementary activity. And you can see the first curve, the one which goes up very, very quickly, is normal immune serum globulin produced for intramuscular use in the old days at a normal pH. You can see that this has a high level of complement activation, whereas preparations such as gamma mu, a preparation of immunoglobulin released for use, has a much lower capacity to activate complement and consume complement in the test system. These tests are again done on each and every batch. I want to now talk about some other characteristics which have been in the past 10 or 15 years shown to be very important because they can also result in adverse events in patients. And you all know that in 2010 we had what one might call a crisis in this sector, whereby several immunoglobulin preparations were associated with thrombosis. Not all, but more than one brand was associated with thrombosis after being administered to patients. And this is a slide which simply shows the incidence and prevalence of this phenomenon in the various patient groups which are given immunoglobulin. You can see that the largest numbers involved immunomodulatory diseases such as CIDP and ITP, but patients with primary immunodeficiency, hypogammaglobulinemia, were also affected. So this was an important and difficult situation for the industry to resolve. But as a result of much investigation, which commendably was done very, very quickly, it was found that it was the procoagulant factor 11 as a contaminant in immunoglobulin preparations, which was responsible for the thrombosis. It is important to note, I think, that this was something which was actually known from 10 years before we started having this massive problem in 2010. This was already significantly measured as can be shown in this publication, but it was not thought to be something which was extremely problematic because at that time thrombotic events were much, much rarer than they ensued in 2010. But it's important to note this, and we must also always be vigilant. When we have observations of this kind, they need to be acted upon immediately and not wait until patients start experiencing adverse events. So factor 11 is an important physiological component of the coagulation system. We need factor 11. Patients who are congenitally deficient in factor 11 bleed. It is, in fact, a form of hemophilia called hemophilia C, very rare, but excessive activation of factor 11 to factor 11A leads to thrombosis, and this is what occurred as a result of the contamination of the immunoglobulin preparations. And so now, again, coming back to our regulatory oversight, we have the requirement that the method of preparation also includes a step to remove thrombotic agents. This step is emphasized as needing to identify activated coagulation factors, including factor 11, but we need to keep in mind that any activation of coagulation in immunoglobulin preparations is undesirable, and other procoagulant agents that could be introduced by the manufacturing process need to be identified and eliminated. And this is something which now the industry needs to do on a routine basis in order to avoid this severe adverse event. And you can see that now we have characterization of the manufacturing process as required by the pharmacopoeia to identify the steps where there is factor 11 and ensure that the process removes them. So here we have an antibody, an immunoglobulin purification process. You can see that there are 11 steps here which are all characterized in relation to their content of factor 11, and you can see that between step 7 and step 8 there is a huge drop as a result of the manufacturing process in the concentration of factor 11, which is in fact undetectable in terms of the threshold of the test. So it is important to note that this is a characterization which is done by all companies now for their immunoglobulin product to assure and ensure that the manufacturing process can eliminate, besides viruses and other pathogens, factor 11 and other procoagulants. It is very important. Another characteristic adverse event which has occurred in the past years has been an increase, all of a sudden, of hemolysis in patients who are given immunoglobulin treatment. Again, this was an adverse event which was known to occur before the sudden increase. However, there was, as with the thrombotic events, a significant increase in the incidence of these events. Here we have another similar slide to what I showed before, showing the patient groups which have suffered this adverse event after being given immunoglobulin, and they include also patients in the community of primary immunodeficiency disorders. We know that this is due to the retention within the immunoglobulin preparation for therapeutic use of excessive amounts of anti-A and anti-B antibodies, which are the antibodies which are associated with blood groups A, B, and O. Usually, the formation of a polyclonal, remember that word, polyclonal plasma pool results in these immunoglobulins being neutralized, forming what we call immune complexes, which are then precipitated out of the product during the purification using ethanol fractionation so that they are diverted, if you like, from the final immunoglobulin solution. Therefore, this was not a problem as long as we had continuous precipitation of these so-called immunocomplexes over the course of fractionation. However, it was evident some years ago that the new generation of immunoglobulin products, which I told you were not being produced using cone fractionation, were retaining some levels of these anti-A and anti-B and causing hemolysis in patients, a severe adverse event. And therefore, we have within the pharmacopoeia and have always had, even before this higher incidence, the measurement of anti-A and anti-B immunoglobulin to ensure that it is not over the threshold associated with adverse events. It is important to note that this process resulted from the transition from traditional ethanol processes to modern processes utilizing immunoglobulin purification, such as ion exchange chromatography, gel filtration, and so on. And this is shown on this slide, in which several steps of this purification scheme here. This is a product you're all familiar with. You can see when this product was produced with, when an immunoglobulin product was produced with cone fractionation, the final product, the bulk solution we call it, had very low levels of anti-A and anti-B hemolysins, whereas the initial purification of a product such as Privigen, but this occurs for all other modern products, did not remove this important contaminant from the final bulk solution. And it is because of this that the company, like other companies, has introduced processes to ensure that the immunoglobulin does not contain these important adverse event causing antibodies. I'm approaching the end of my talk. I was asked to touch upon a couple of recent occurrences which are of interest to this community. Recently, a group has proposed an immunoglobulin solution for patients in less economically rich countries, low and middle income countries, such as Ethiopia, using a fractionation scheme which is different from those which are used to produce the products which I have been discussing with you. This is described in this publication, and this is what we call, if you like, a do-it-yourself immunoglobulin, which can be done within a normal blood bank to produce an immunoglobulin solution using this scheme here. It is no secret that IPOPI, and I have myself been involved in this, has severe reservations about this scheme because of some features of this manufacture which run counter to the principles which I have been describing to you. Particularly, this is a small pool product. It does not adhere to the requirement of having 1,000 at least blood donors represented, and therefore for patients with immune deficiency, we run the risk of not having the full spectrum of therapeutic action which is needed within the immunoglobulin. This is a product which needs to be stored frozen. It needs to be stored deep frozen. It cannot be stored in a normal refrigerator or even at room temperature, as some products nowadays can be stored. And very, very importantly, within this fractionation scheme, we do not have viral inactivation for so-called non-enveloped viruses. I shall say something about this in a moment. But this scheme has not shown, publicly at least, any viral inactivation for non-enveloped viruses, which we think is something of severe concern for patients who are going to be receiving this product on a very long-term basis. Viral inactivation, as I said, is a requirement by the regulatory authorities. On this slide, I simply show an extract of the FDA's requirements, which says that there have to be at least 2 viral clearance or inactivation steps. And because of the difficulty of removing non-enveloped viruses, at least 1 removal step is expected to be included in the manufacturing process, which can remove these non-enveloped viruses. And as I intimated previously, this is at the moment absent in the do-it-yourself scheme proposed by the group. You can see that other immunoglobulins, such as those we have on the market, again, I have here chosen one particular preparation because it is well described in public information, but this is the case for all approved products nowadays in America and Europe. And you can see that in these viral activation schemes characterizing the whole of the immunoglobulin process, including the viral elimination schemes, such as virus filtration and low pH, you can see that the parvovirus B19 and other types of non-enveloped viruses are very effectively removed. And this is a very important point, and it is required to conform for the regulatory authorities. So you can rest assured that any immunoglobulin which is available for the market in Europe and North America has these requirements to the full. I want to make one final point because I'm aware, and I have been made aware, that IPOPI itself has been approached by certain companies and individuals claiming that they have novel technologies which are of benefit in ensuring higher yields of immunoglobulin and therefore access to patients. I do not wish to go into this issue in detail, but I have summarized my thoughts on this in this slide. These are claims which have been made by certain new technologies. There are hardly any details supporting these claims in public information. However, in public information, statements made by certain companies regarding the history and status of established technologies are inaccurate. For example, the statement, as I said before, that companies in the traditional plasma fractionation scheme have not innovated their treatments for 80 years. This is totally not the case. These groups which are making superior claims regarding the safety and efficacy of their technologies, they are unsupported by evidence. And in this presentation, which I have made to you today, I have tried to emphasize safety and quality issues, which should be foremost in all your minds if you, as representatives of the patient communities in your countries, are approached by people such as those who make these claims. And I encourage you to have great caution in approaching and reacting to organizations making such claims. I just want to draw your attention to this book, which is available on the IPOPI website. It is a book which I have written for IPOPI, and I have been involved in this project for quite some years now. The current edition is the 2nd edition. It is available on the website. It is from 2018. And at the moment, I am working on a 3rd edition, which I think will be available certainly before the end of this year. I thank you very much for your attention. And again, I express my regret that I have not been with you today.